Let's discuss hemoglobin derivatives. Hemoglobin breakdown products include porphyrins, bilirubin, and urobilinogen. Step 1. Hemoglobin breakdown. Hemoglobin from red blood cells is broken down into a substance called verdohemoglobin. This marks the very beginning of the metabolic pathway. Step 2. Formation of biliverdin. Verdohemoglobin is further processed into biliverdin, along with iron and globin. The iron is stored for reuse, while the globin proteins are recycled. Step 3. Conversion to bilirubin and transport. Biliverdin is reduced to bilirubin. In the blood, bilirubin binds to albumin, and this bilirubin-albumin complex is transported directly to the liver. Step 4. Conjugation in the liver. Once inside the liver, bilirubin undergoes conjugation. This process attaches glucuronic acid to form bilirubin diglucuronide. Conjugated bilirubin is water-soluble, which is important for its excretion. Step 5. Normal bilirubin metabolism. Finally, conjugated bilirubin enters normal metabolism. It is excreted in bile and continues through the pathways that are studied in urinalysis and body fluid testing. Let's review the laboratory analysis of bilirubin. First, let's begin with the diazotization methods. In the classic method, bilirubin reacts with diazotized sulfonylic acid to form a purple compound called azobilirubin. Total bilirubin, which includes both conjugated and unconjugated forms, reacts slowly with the diazo reagent. On the other hand, conjugated bilirubin, which we also call direct bilirubin, reacts rapidly in water. The Jendrassik Grof method improves this reaction by using caffeine benzoate as an accelerator. Remember that direct bilirubin is conjugated and water-soluble, while indirect bilirubin is unconjugated and relatively insoluble. Next, we move to the direct spectrophotometric method. This technique is mainly used in newborns. Why? Because newborns lack many of the interfering compounds found in adults, making this method effective for them. Now let's talk about specimen collection and handling. Bilirubin is light-sensitive, so samples must always be protected from light. If exposed, results may be falsely low. Lipemia, which means excess fat in the sample, can cause a false increase in results. Hemolysis may cause either a false increase or decrease, depending on the concentration of both hemoglobin and bilirubin present. Finally, let's cover some general information. When unconjugated bilirubin levels are elevated, urine urobilinogen also increases, because more bilirubin is reabsorbed from the intestine and filtered by the kidneys. When conjugated bilirubin, which is water-soluble, increases, it appears directly in the urine. Let's talk about jaundice. Pre-hepatic jaundice occurs when there is increased destruction of red blood cells. This leads to a rise in unconjugated bilirubin. The liver function itself is normal, so conjugation continues at the usual rate. Because of this, conjugated bilirubin remains within the normal range, and there is no bilirubin detected in urine. However, the excess unconjugated bilirubin results in an increase in urine urobilinogen. Hepatic jaundice, as seen in viral hepatitis or cirrhosis, involves dysfunction within the liver. Both unconjugated and conjugated bilirubin increase, along with urobilinogen. Because conjugated bilirubin is water-soluble, its elevation causes bilirubin to appear in the urine. At the same time, urine urobilinogen also rises. Post-hepatic or obstructive jaundice occurs when bile flow is blocked. In this case, both conjugated and unconjugated bilirubin back up into the plasma because they cannot be metabolized properly. The blockage prevents conjugated bilirubin from reaching the intestine, which means urobilinogen levels decrease. Since there is less urobilin formation, stools may appear clay-colored. Let's further review bilirubin and disease states. Pre-hepatic jaundice, such as in hemolytic anemia. When there is increased destruction of red blood cells, more unconjugated bilirubin builds up in the plasma. Conjugated bilirubin remains normal because the liver is functioning properly. In the urine, there is no bilirubin present since unconjugated bilirubin is not water-soluble. But urobilinogen is increased due to the excess bilirubin being converted in the intestine and reabsorbed. Hepatic jaundice, as seen in cirrhosis or viral hepatitis. Here, the liver is damaged and cannot process bilirubin efficiently. Both unconjugated and conjugated bilirubin levels in plasma are elevated. In the urine, bilirubin may be absent or increased depending on severity. Urobilinogen is increased as well due to impaired hepatic processing and increased leakage into circulation. Post-hepatic jaundice, also called obstructive jaundice. This occurs when bile ducts are blocked and bilirubin cannot reach the intestine. Plasma will show normal unconjugated bilirubin but elevated conjugated bilirubin. In urine, bilirubin is increased because conjugated bilirubin is water-soluble and backs up into circulation. Urobilinogen, however, is decreased since conjugated bilirubin cannot reach the intestine to be converted. During pregnancy, several important laboratory tests are performed to monitor both maternal health and fetal development. At around 16 weeks of gestation, maternal serum is analyzed. One test looks for neural tube defects such as spina bifida by measuring alpha-fetoprotein, also known as AFP. Another screening checks for Down syndrome. This can be done using a triple screen, which measures human chorionic gonadotropin, AFP, and unconjugated estriol. An expanded version, called the quad screen, adds inhibin A. Amniotic fluid is also used in testing. One purpose is to detect hemolytic disease of the newborn, most often caused by Rh isoimmunization. In this case, bilirubin levels are measured spectrophotometrically at 450 nanometers. Another use of amniotic fluid testing is to assess fetal lung maturity. This is often done by calculating the lecithin to sphingomyelin ratio. Vaginal swabs can help predict premature delivery by detecting fetal fibronectin, a protein secreted at the boundary of the amniotic sac and uterus. A negative test result is reassuring, as it indicates that preterm delivery is not expected within the next 2 weeks. Another application is testing for premature rupture of membranes. The AmniSure test detects amniotic fluid protein known as PAMG1, which appears in cervicovaginal secretions after rupture. After birth, newborns are screened using dried blood spot specimens. These samples are analyzed with techniques such as tandem mass spectrometry, fluorescent immunoassays, electrophoresis, and real-time PCR. Newborn screening identifies more than 30 genetic and metabolic conditions, including phenylketonuria, congenital hypothyroidism, galactosemia, cystic fibrosis, sickle cell disease, and other metabolic disorders. Let's walk through hemolytic disease of the newborn and the role of bilirubin in neonates. First, in newborns, it is normal for indirect or unconjugated bilirubin to be elevated. Conjugated bilirubin, however, usually remains within the normal range. Second, because unconjugated bilirubin is water-insoluble, it cannot be excreted into the urine. That means there will be no urinary bilirubin detected in neonates. Third, unconjugated bilirubin also cannot be broken down by intestinal bacteria. This results in the absence of urinary urobilinogen. On a dipstick test, this appears as normal. However, it is important to note that this pattern is different from other hemolytic processes. In those cases, urinary urobilinogen is typically increased. This sequence highlights the unique bilirubin metabolism in newborns with hemolytic disease.